All right, I'm going to go ahead and get started. So I want to welcome everybody to um, a sip of science. So that it's the series of presentations that we do from the Biodesign Institute every year to celebrate some of the research that's ongoing here at Arizona State University. I'm supposed to give you some housekeeping notes. I think the most important of which is that after Hao gives his presentation, we're going to have questions. And as you can see here on the image at the bottom of the screen, you should see the word chat. Just click on that link and type your question into the chat, and then someone will ask that question so that we can, that allows us to, if several people ask the same question, it allows us to sort of combine them all. We'd also appreciate if everybody could mute their microphone in the lower left part of the screen during the presentation. And if you want us to read your name a different way than it's showing on the screen, you can rename yourself by clicking rename on your screen and changing it. That's up to you all. All right, so let me just briefly mention that we are all here from the Biodesign Institute. This is the premier research institute at Arizona State University. Our institute is focused on translational research. That is to say, we don't do research for the sake of research. We do research to solve world problems. And because of that, we are organized into centers, and each center in the institute is focusing on one problem or another. And within each center, we often have faculty from all over the university, because you don't solve problems with everybody looking alike. You solve problems by bringing together scientists with different backgrounds and different skill sets, each one approaching the problem from a slightly different angle so that they can solve the problem. And so that's the whole concept behind our institute. We now have 17 centers and about 130 different faculty members at this point. And you will find research at the institute ranging from cancer research to, as you're going to hear, nanotechnology today, people doing cybersecurity, people converting toxic compounds in water into reclaimable metals, while at the same time cleaning up the water to make it safe again. So broad range of research within the Institute. And hopefully, as you attend some of these science meetings, you'll learn more about all of the really cool stuff that we're doing. I will mention briefly that the Institute played a big role in responding to COVID-19 this last year. As many of you know, we developed the saliva test, and we are rapidly approaching having done around 1 million tests now throughout the state of Arizona, with a current focus on trying to reach underserved areas to get testing in the context of the current Delta wave that we're facing here in the country. Okay, so let me tell you a little bit about Sip of Science. Sip of Science is what you're at right now. This is a program where we have some of our top researchers from the Institute come and talk to you about what they are working on, the ideas behind what they're working on, the approach that they've used to solve the problems that they're solving, and what they view all of that work means. It's really meant to be a conversation, and so as you'll see, it's a relatively short presentation, followed by a decent amount of time so you can ask questions and really we can hold a discussion about what's going on. In the perfect world, back when this started, we would meet at a pub or at a restaurant where we would have literally a sip, we would have a drink of something, and we would have some food to eat. And in fact, we were hoping to go to Olive and Ivy this year at this particular meeting, and we thank them for having planned that with us. Unfortunately, the Delta wave has sort of taken over. There's just a lot of COVID going around right now. And we heard from a number of you that you weren't feeling very comfortable about meeting in person just yet. So we will, in the spring, get back to Olive and Ivy. We're looking forward to getting over there and having a in-person seminar there. But for the time being, A Sip of Science is going to stay virtual right now while it's a little bit safer. So I think that those are the primary comments I had to make at the beginning here. So now it's my pleasure to introduce tonight's speaker, who is a friend and a brilliant scientist. The word genius comes to mind. He directs the Biodesign Center for Molecular Design and Biomimetics, and he's a professor in the same school. I'm a professor in the School of Molecular Sciences. he does all kinds of very creative research where he has to, in his head, sort of think about three-dimensional structures at a scale well below anything that any eye could ever see. So he's imagining how molecules assemble themselves into different shapes and how to use those different shapes to accomplish different tasks. So creating essentially nanobots to do all kinds of cool stuff. I will also mention to you, because I've traveled with Dr. Yan, that he's also quite a good singer. He also plays guitar, and he's really good at karaoke. So not sure if he's going to plan on doing any of that tonight. But in any case, let's welcome the Milton D. Glick Distinguished Professor in Chemistry and Biochemistry, and he'll start sharing his slides and tell you about his work.
Thank you, Josh, for the introduction. And it's my great honor to present today's topic. And thank you for joining us for the sake of science tonight. Let me open my slides. Can you all see my slides?
Yep, looks good.
Today, I'd like to talk about, discuss this topic of tumor-attacking nanorobots, and want to set the stage to discuss whether this is very futuristic fiction or is a real thing coming in the next few years or next 10, 20 years. As Josh mentioned, I'm leading the Biodesign Center for Molecular Design and Biomimetics. What we do here in the center is to use bio-inspired approach to use biomolecules to create very small things at the nanoscale and to address many of the challenges for, including the medical challenges I'm going to talk about today. Here is the problem. Everybody knows that cancer is one of the major leading cause of death worldwide. In 2018, there were about 18.1 million new cases and 9.5 million cancer-related deaths worldwide. By 2040, it was predicted that the number of new cancer cases per year is expected to rise to 29.5 million, and the number of cancer-related deaths to 16.4 million. And this can cause a lot of problems for the economy. The Agency for Healthcare Research and Quality estimate that the direct medical cost for cancer in the US in 2015, it was like 80.2 billion US dollar, and it's quickly coming bigger and bigger. So the question is, can we find a cure for this problem? People have been trying different ways. Scientists and doctors are coming together to develop different ways to treat cancer, including those invasive methods like surgery, radiotherapy, and also like non-invasive methods to cure cancer, including chemotherapy and other methods to deliver the drug payloads to the cancer and tumor site. So let me see, the arrow doesn't work. So there are among many challenges in cancer treatment. One of the main challenge is there's off target. The drugs we use does not always go to the right site to kill the tumor, and which is also a major cause for the side effects. And as you know, many of the chemical drugs will not only kill the tumor, but also will kill the normal healthy cells. And another issue is the drug resistance. Over time, the body will evolve and develop ways to against the drugs. So some chemical drugs will not be useful after long term. And another thing is the metastasis. And we know that the tumor will migrate before we find it and kill it. So even we can find a solution to tackle one of the problems, that will be significant and can even improve the strategies for the cancer therapy. Wouldn't it be wonderful if one day we can inject some molecular robots to go into the bloodstream and only target at those tumor sites and kill the tumor. In 1966, there is a movie called Fantastic Voyage, and it was a science fiction movie trying to imagine that we can send into the bloodstream a submarine crew to go to a scientist's brain to repair the brain damage. So this is, of course, a fiction, but it has inspired generations of scientists, including myself, to find a solution to be able to create a robotic system that at very tiny scale that can go into the bloodstream and to cure cancer. So this is an animation from Boston. This is a reality, and people now can invent and produce robots of a macro scale that can respond to music, can do certain tasks of carrying different loads. But according to the website of Boston Dynamic, this took like 30 years of a development from the concept to the realization of such a kind of a robots of doing fancy things. So the question is, can we go very small with this kind of robotic actions? So how small can we do? If you look at nature, I want to talk about the scale of nature. And the human has a size of 1 meter to 2 meter. And we know that each cell is about 10 micrometer. And the width of a human hair is about 100 micrometer. If you go down to the size of the virus, like a coronavirus, the size of the virus is about 100 nanometer. So it's 1/1000th of width of the human hair. If you go down further, a protein molecule is about 10 nanometers, and the width of the diameter of a DNA molecule, which is the genetic molecule of the body, it is about 2 nanometers. So by the way, 1 nanometer is about 1/10000000th of a meter. So it's very tiny, small scale. We cannot see with the naked eye. We have to use those electron microscope, different nanoscopy to really visualize those small molecules. So, and if you look at nature, if you look at biology, biology is full of robots. Including those motors that can replicate DNA, called the DNA polymerase, and those motors that can move and guide the cells to different chemical environment. So here I would like to show one animation illustrating there is a 2-legged molecular motor carrying a bag of newly synthesized proteins to transport this newly synthesized proteins from one side of the cell to a different location on the highway track inside of the cell. Molecules from one side to the other side along a highway track called microtubule. And this highway track is a self-assembled protein molecules into such a long tube. And this 2-legged motor can walk on such a highway track with a very high speed. So the question is, can we design molecular machines or molecular robots from scratch, from the first principle? So to do this, we got inspired from nature, from biology again. If you look at the biology doesn't form protein molecules by manipulating the atoms one by one and put them together into a functional molecular machine. Biology forms a functional molecular machines through self-assembly, through molecular self-assembly. And this kind of a self-assembly is instructed by the genetic information. So basically, if you look at the biology, we have the genetic information buried, embedded in the DNA. And the DNA is used to transcribe into a molecule called RNA, and RNA will serve as an intermediate and get translated into a protein molecule. And the protein molecule will be the functional molecule that can catalyze many of the reactions, can cause movement, like the motor proteins, two-leg motor proteins I just showed you in the last slide. So biology uses self-assembly to form molecular machines, molecular robotics. So here I would like to tell you an idea of how do we fold a biomolecule into a different nanostructures. So this is a strategy inspired by the origami. You might have heard about the Japanese paper origami. Is a way to fold a piece of paper into different three-dimensional shapes of different kinds of artistic views. So here we use DNA instead of a paper to fold this DNA molecule, a long piece of a DNA molecule, into different kinds of nanoforms at nanoscale. So I'd like to show you one animation. So imagine that we can take a single-stranded DNA, which has a long strand of a DNA containing different sequences, different base pairs, A, T, G, C base pairs. You learned this from your kindergarten. And then we can add into other DNA molecules called a staple DNA, staple molecule. And those staple DNA can fold and bind different segments of this long piece of DNA and fold this DNA into a different geometry. So basically here we have a way to custom make a different DNA molecules through a self-assembly. So folding a DNA into different geometry through self-assembly. So I'll give you several examples of such a design. So here you're looking at a DNA origami design on computer. And you can use a computer. We have developed softwares to allow us to develop different geometries of the DNA folded by the DNA sequence. So here is an example. It is one design. You're looking at the flowers with a hummingbird, and the hummingbird is attaching to the flowers. And then we can synthesize the DNA molecules and add into a tiny little test tube, and then allow the DNAs to find itself and self-assemble into such kind of a design. What you're looking at here is an atomic force microscope. It's kind of a microscope that can allow us to visualize the self-assembled product under the microscope to reveal the detailed structures, design structures at nanometer scale. So this is the real experimental result. You're looking at here, you see the flowers and also the hummingbird. So basically, if you design something, you can allow the DNA to fold into this type of a structure. And people from the field of DNA nanotechnology, DNA origami artists, like molecular engineers, have been able to create a different kind of strategies to fold DNA into different forms, including the smiley face, different alphabets, different characters and letters, and the shape of a human, and also the Mona Lisa, smiley face of a Mona Lisa. So all of this illustrated here are all real experimental results. It's not artistic drawing. And you can see that we can use DNA to fold into different architectures. All right. So one beauty of this kind of a strategy of self-assembly is that simultaneously in a tiny little test tube, I can have a smiley, 1 billion, trillions of molecules of the smiley face and Mona Lisa, and also the 26 letters of alphabets. All right, so this illustrates the power of folding DNA using this strategy called DNA origami. So at this point, you might start to ask, why do we want to fold this, what can we use this for? So here it comes to the idea of using this to create molecular machines, a molecular robots to kill cancer. So we would like to fold a molecule, and this molecule can sense the local environment in the body and compute and decide whether I want to kill the cancer or not. So this is the cartoon to illustrate that what we like to do is to develop a nanorobot that can be injected into the bloodstream and allow the nanorobot to target only to the tumor site and do the job to kill the tumor only at the local tumor region. So I'd like to explain the concept and the idea and using an animation. So basically, what we do here is to use the DNA to fold into a planar structure, showing here. And then we can attach some drug payload, a cargo molecule called a thrombin. Thrombin is one kind of a protein molecule in the cell responsible for the blood coagulation, blood clotting. And it will trigger a polymerization process to allow the blood to stop bleeding. But if you just inject the thrombin protein into the blood, it will cause blood coagulation everywhere. So basically, we will just die, right? And even before it reaches to the tumor site. So what we do here is to fold this piece of a DNA nanostructure into a cage to hide the thrombin protein inside of the cage until it reaches to the tumor site. And we lock the cage with a molecule. Now, only when it go to the tumor site, circulate in the blood and go to the tumor site and respond to a K molecule, this K molecule only got produced in the tumor. It's called overexpression of a certain molecule at the tumor site, and now it can unlock the key and unlock the lock and open up the cage, and then expose the thrombin protein to cause the blood coagulation only at the tumor site. So once the blood got coagulated, and it will stop the nutrients to the tumor and stop out the tumor, so the tumor will eventually die. So this is the whole concept of this nanorobot system. So here's the animation showing that we can fold, the gray thing here is the DNA nanostructure, and we allow it to fold into a cage to protect the thrombin, the drug payload. And then we lock it and inject into the blood and allow it to circulate in the blood, because there is a lock molecule which also serves as the targeting molecule. It will reach only to the tumor site, and then it will get opened up to expose the thrombin molecule and will trigger the blood coagulation only locally at the tumor site. And then it will stop the nutrients to the tumor, and the tumor will eventually shrink. So here is just one piece of data, and this work has been published in Nature Biotechnology. And it's a work in collaboration with Chinese Academy of Sciences. And here you can see that the nanobot can go to the tumor site and effectively only target the tumor and shrink the tumor. So at the bottom row are the shrink the tumor compared to some of the control experiments without the nanobot. And with the nanobot, and we can significantly increase the survival rate of the patient, and also using a model system, and also shrink the tumor effectively. So there are also different ways of designing a nanobot system, and many people are interested in the idea. And our vision here is to design a designer. So I'm in a Center for Molecular Design and Biomimetics. We take a biomimetic approach to design a nanorobot system, that can sense and compute and actuate to address some of the medical challenges, including healing cancer or treatment of the and vaccine development of COVID-19. So here I'd like to emphasize what is called sense, compute, and actuate. So sense means that I can use the nanobot to only to sense what is the disease site. And when the nanobot look at the disease, it can recognize some labels on the disease and decide I can implement some molecular computing into the nanobot to allow it to compute and decide whether I can open up and release the drug. So this is the thing called sense, compute, and actuate. And this is a long way to go, but we are in the process of developing an intelligent nanobot system that can help address many of the medical challenges in our human society. With that, I want to thank you and take any questions you may have about the system we're building. Thank you.
Very cool. Okay, well, thank you, Hao. All right, so I think we're gonna, turn off the screen and we're gonna open up the floor for questions. It looks like there's already some things in the chat there. So I'm gonna hand this over to Julie, who's gonna be the question asker today.
Right, so Julie Liss asks, what type of tumor is being shown in the previous slide? And besides thrombin, can it also carry chemotherapy to the tumor site?
Yeah, we tested using four types of tumor models, including lung cancer and prostate cancer, melanoma, and ovarian cancer. And all of them show the very effective tumor killing, like a shrink of the tumor using the nanobot system. And the thrombin is just one type of drug payload we can try. Right now, we are using, you can imagine we can load chemo drugs and also siRNA, some antisense oligos that can be used to silence the genes and kill the tumor, and with different kind of a strategy.
Kirk from Phoenix asked a related question. Are there different types of tumors that the nanobot would not work for?
Right now we are developing, looking for some like a different targeting mechanisms. And hopefully it will be more specific to certain tumors. When some tumors develop different biomarkers, express different proteins, we would like to use this ligand or targeting molecules to allow us to target different tumors. Right now, we only tested 4 kinds of a tumor, but we're in the process of testing on other kind of tumors. But this is only the proof of concept. There's a long way to go. But it's quite exciting to be able to see we can put some intelligent design into a self-assembled molecule that can go into the bloodstream and do the work.
So Jim Kalin asks what human trials are being planned and scheduled. And so I think we all are interested in, when might we see therapies like this available?
That's why we need a lot of funding support to move this technology further. And I think it will take a few more years, 5 to 10 years, to move to the clinical trial. So right now we're focusing on loading. Thrombin is just a model system, right? Thrombin may not be the safest, right? There might be leakage and unexpected leakage to expose the thrombin unexpectedly when the nanobot is circulating. So we are working on, for example, loading siRNA to the system and investigate whether it will work or not. I anticipate it will take a few more years to go from the research to preclinical trial and clinical trial. We did form a company called Nanobot to tackle some of the problems that might help facilitate the research agenda. Thank you.
How does the nanobot self-destruct, or does it remain passive after the tumor is destroyed?
Over time, you can imagine this is DNA is a biomolecule. It will get degraded over longer term, get cleared out.
Michael envisions a time when people will just ask every year to go and routinely be injected so that silent cancers can be attacked proactively. So I think you've got one person ready for ready for it.
The people asking me, emailing me, saying, "Oh, we want to, yeah, can we receive the drug and the nanobot? We want to try." They're very desperate, but unfortunately, sadly, I have to respond to them saying that this is still lab research. It will take a few more years to really push that to a clinic.
Cade asked a really interesting question. So would nanotechnology be a cheaper form of treatment for cancer generally? I know you may not be the cost guy, but
we did, I can't say for sure for other nanotechnology, like people have been using nanoparticles, lipid nanoparticles. For example, lipid nanoparticle, people use this for the current COVID vaccine, right? So cost is not a major issue, I assume, but people have to pay for it. But we did do a calculation of how much the DNA cost, whether the DNA is very costly. And people imagine that DNA is very costly. Actually, if you look at the calculation, is not that expensive, right? 1 gram of a DNA with current synthesis method can be controlled to less than $1. Yeah. But with for technology, people will come up with the ideas to scale down the cost to make it much cheaper to use.
how do you avoid the cage structure from disassembling in the bloodstream?
There are different strategies. You can chemically modify the structure to make it stable. A natural DNA is not as stable as chemically modified DNA, and because the enzymes inside of the cell, the proteins, can chop up the DNA. But there are different strategies to avoid it that need to be tested in the lab.
How about transporting targeted CRISPR technology to specific cells? Is that something that has ever crossed your mind?
Yeah, I mean, we are collaborating with some people. Right now, we're thinking about using this nanobot as a delivery vehicle to deliver CRISPR-Cas9 system. Yeah. It's something very interesting. It's a good direction to think about.
Let's see what else. Have you looked at nanobots finding and releasing biomarker or scanner chemicals at cancer cell sites, perhaps years earlier than treatment chemicals?
We haven't tried any chemical drugs, chemotherapy drugs in our system, so we don't know exactly whether this. So small molecules is another target we want to try, but we need to seal it better so it doesn't leak out to the bloodstream and kill the normal cells.
And Julie Liss asked another question. Had you envisioned the targeted neurodegenerative disease technology with crossing the blood-brain barrier into neurodegenerative disease targeting?
Right now, not yet. There's like a blood-brain barrier problem issue, and the size of the particles we're using might be too big. But I know there like some research is developing like a modified DNA that can bypass the blood-brain barrier. So something to keep in mind. Thank you, Julie.
Peter Williams asks, cancer is very heterogeneous, right? Have you identified a universal cell surface marker?
The cell marker we're using here, the key molecule, is the nucleoline, is universally overexpressed in all the cancers. So that's the thing we're using. There's an elevated concentration of the nucleoline. But there might be, this might be a good question for Josh. He's a real expert in the cancer marker.
Well, actually, I was going to turn it around to you and say, talk a little bit about how you might target these things in other cell types, because you could imagine cancer is one treatment, but there's lots of other diseases where targeted delivery could be very helpful.
That's true.
You guys thought about targeting other cell types, and how would you go about that?
For example, we are developing a transmembrane sensor for intracellular targeting, right? So this little tweezer, as a molecular tweezer, will contain a piece of a probe to only detect intracellular and messenger RNA. And then you can imagine that it was floating around in the cell, and only when it recognizes a piece of mRNA overexpressed in certain cancer type, and it will trigger a conformational change, and that can cause a cascading event to allow the nanobot to open up and release the payload. So relying on, Josh, I'm not sure if you agree or not, relying on protein markers might be difficult. So,
yeah, it is difficult. People have tried for years, and they're not reliable enough.
Yeah, so I would say, but this is something we are developing, a transmembrane sensor that can intracellular target.
There might be some unique cell types, like very specific organs that have, unique markers to them, kidney or something like that, where you could get away with it. But yeah, it's not easy.
That's a challenge.
Yeah.
Did you want to talk a little bit more about the sensing that you also have in mind? So how would that work in a disease therapy, for example?
Yeah, I mean, the sensing, compute and actuate, that's the dream project, right? So, I mean, there are, like, for example, sensing a different pH in the tumor microenvironment on mostly at the lower pH. So if we can have a nanobot that can contain the lock molecule that can enfold, for example, the I motif sequence that can react with low pH, local low pH, and enfold to open up the nanobot to release the drug. So that's the one thing. We just talk about different biomarkers. What we like to do here is to, for example, creating multiple locks, multiple targeting, and only when all the biomarkers are present and on the surface of the tumor cell, it can unlock the nanobot, unlock the cage, and release the drug. So that's the computing aspect, right? So basically, it will be a molecular computer that can sense and the biomarker, and then decide whether I want to open it to accurately, precisely release the drug to the tumor.
Okay, let's see. And how early on in cancer progression can nanobots detect the mass?
As early as possible, if we can use the bot. So the one idea is to develop a theranostic, right? So basically, it comes back to the sensing, right? If we can have enough of the tumor cell that can recognize, and maybe we need some amplification and to recognize a small amount of the tumor at early stage, and amplify the signal to trigger and attract a more nanobot to the site. Another way is to think is to allow us to a nanobot to use for immunotherapy. right, to allow when the tumor is at early stage. And what about developing a nanobot containing some immunotherapy molecules or adjuvants to trigger our own immune system to be able to attack the tumor at the early stage. We have some ideas in collaboration with an immunologist to work on that.
Jim asked an interesting question too. Can a nanobot carry a large payload of sensitized antibodies or T cells to a cancer cell? It wouldn't be toxic to non-cancer cells. Yeah.
So I don't think the nanobot we are developing can hide the T cell. The T cell is much bigger than the nanobot, but you can imagine that we can create a platform with a double ligand, one side with a molecule that can bind to the T cell and recruit to the cancer cell on the other side. So this will be a bridge to bridge T cell immune cells to the cancer cells, but with some trigger molecules.
And I have a question. So how many other universities or research groups that you know of are working on similar kinds of technologies?
Well, people from Harvard to Caltech and many labs are working on the same objective. And, we're one of the, Biodesign Center for Molecular Design and Biomimetics is one of the major center of research working on this type of problems. We're very proud of having several really brilliant young faculty working together and interdisciplinary to tackle the problem. I would say Biodesign and our center is one of the probably I'm to say one of the best in the world on this topic.
And do you ever hear about safety concerns? What if we could lose control over something like this? What do you say about that?
Yeah, the people always ask, right? Well, there's a gray goo problem that the nanobot can go very wild and replicate in the body. If the nanobot system we develop is really a simple, right? It's just one sensing and one opening, and the structure we have cannot replicate, right? And because of the structures we make and the staples going from one segment to another segment, and the DNA polymerase just cannot recognize it.
Right. Right.
And it cannot go through all these junctions to make more of the nanobot, right? And also, the DNA is more of a biocompatible molecule. It's borrowed from biology and giving it back to biology. I think it will be an ideal molecule if we want to develop a nanobot. Instead of using gold or silver or other nanoparticles to go back into the body. But of course, we always be very cautious of designing the sequence to make sure that those sequences that we used will not get break down and get incorporated into the genome. And because of the size of the nanobot, it will not go through the nucleus membranes, right? So it will stay in the cytoplasm. So it's going to be harder to incorporate in the genes.
Can the technology be used as a diagnostic to detect cancer?
Yeah, as I told you that we are developing a transmembrane actuation and a sensor system using DNA nanotechnology that potentially can be used for diagnosis.
Okay.
Is that sensor something that will be coupled with an imaging system so that you could locate?
So you can imagine that inside we have a probe, outside we can have amplification to allow it to, once it trigger from open to close or close to open, it will expose the trigger molecule to cause hybridization chain reaction or amplification to allow us to image.
That's great. Yeah.
We should talk about, discuss this.
Yeah. Locating cancers is often one of the biggest challenges. It's equally frustrating to know that there's a cancer there, but you can't find it, right? So a tool that would help us find it would be very cool. All right.
I think we have time for, you want to just ask one more question? Is that okay, Josh?
Okay, one more. Yep.
Okay. Jim asked, can a swarm of nanobots adhere to the surface of a cancer cell and obstruct its access to nutrients and gases?
Can you repeat the question one more time?
Can a swarm of nanobots adhere to the surface of a cancer cell and obstruct its access to nutrients and gases? So it obstructs blood, right? So, but could it obstruct other things like nutrients and gases to the cancer?
Oh, that's the idea, right? Here is like stop the blood supply. The blood supply contains the oxygen, right? To block the oxygen, but you can imagine other ways of maybe on the surface exposing a molecule that can react with certain nutrients and convert the nutrients into other products, right? This need a lot of development of chemistry. So we are not there yet.
And also the number of nanobots you'd need to completely surround a cell would be probably a lot to deliver.
That's true. That's true.
Especially for a cancer, yeah.
I think that's why we use an enzyme, right? Enzyme can turn over and
exactly, yeah.
Some kind of enzyme. People are developing artificial enzymes, can convert one substrate molecule to another molecule. So with the protein design, they might be able to. We have somebody in the center working on protein design problems.
Excellent. All right. Well, I think that was an excellent presentation. We all want to thank Hao. I know we can't clap in real life, but I think everyone is doing so in their own homes there. So and thanks to the people who organized this whole meeting. Carrie Robinson and Julie that you heard from, and a number of others who contributed to organizing this whole event. I do want to mention that we have an upcoming event on October 6th. Jeff Cordover, he is one of the world's leading experts on neurodegenerative diseases. That's been a topic of great interest at SIP of Science in the past, and he will be presenting how he and his team have been managing the disease. He's led the efforts in many ways that has helped solve some of the major problems in Parkinson's disease. But he's also working on Alzheimer's and other diseases, so you'll want to hear that. And then on November 2nd, we have an amazing Arnson Grand Challenges lecture from Zach Rogers. Zach is a longtime leader in supply chain, and if you've been at all following what's happening in the world of business, let's say you went to try to buy a car, for example, and couldn't get one because they don't have chips for the car. You'll know what we're talking about in terms of supply chain problems right now. Or if at any point during the early part of the pandemic, you had to get toilet paper, you probably remember supply chain problems. So he's going to talk about major challenges facing planet Earth on that. And then I'm going to hand this over to Elizabeth Munkle, who works in our office. So Elizabeth, you can take the floor.
Thank you, Josh. As Josh mentioned, my name is Elizabeth Munkle, and I have the great pleasure of working with the Biodesign Institute on behalf of the ASU Foundation. Tonight, you got a glimpse of the amazing work taking place every day at Biodesign. We would love for you to join us in making this work possible. One way to accomplish this is to join our Pioneers Circle. The Pioneers Circle is comprised of science enthusiasts, curious knowledge seekers, and committed philanthropic supporters who invest in Biodesign's groundbreaking research. And there are several on the call tonight, so thank you. Your gift of $1,000 or more each year empowers our teams to take on the most pressing challenges to our health and environment. Philanthropy plays a unique and vital role in our work by allowing our researchers to explore new and untested frontiers of disease detection, drug therapy, and sustainable materials. To learn more or to join, please contact me or visit the website in the chat box. Thank you.
Thank you so much. All right. Well, I think that that covers all of the big stuff. We'll see you guys all on October 6th to learn about the brain. Thank you all for attending tonight. Have a pleasant evening tonight, and I'll look forward to seeing you all in a month. So take care, everybody.
Thanks all.
Bye.
Good night. Thanks for joining.
Okay.